A closer look at mutations. For my 5th genetics video, I will be explaining some of the types of point mutations that can occur and their effect, if any, on the amino acid sequence. I will provide some analogies to baking to help us understand mutations. This is our original DNA sequence, free of mutations. You can see that I've labeled our start and stop codons with green and red highlight respectively. The bottom strand is the coding strand and the top strand is the template strand with our transcribed mRNA. In the middle, I've already included the 5 amino acid chain coded for by the mRNA. This is our original recipe of how to make the amino acid chain and the proteins they are eventually responsible for. Let's take a look at 4 different mutations. Take a close look at codon sequence AGG. If a G became a C, this would code for serine instead of arginine amino acid. This is called a missense mutation, specifically a transversion because the base mutates from a purine G to a pyrimidine C. In baking terms, this would be like using almond extract instead of vanilla extract. The cake would taste different, but not necessarily good or bad. If ACU codon sequence mutated to become ACC, this codes for the same amino acid. So it is called a silent transition mutation because it goes from a pyrimidine U to another pyrimidine C. This is like if you were out of eggs in your kitchen, so you substitute with applesauce. It still makes the same cake. If AGA were to become UGA, this would code for a stop codon, ending the sequence early, which is a nonsense mutation with transition bases as it mutates from AGA to UGA. In our baking analogy, this would be equivalent to not baking our cake long enough, resulting in an undercooked, inedible nonsense cake. Stop codons are essential in ending the amino acid sequence. So if our UGA stop codon became UGC and codes for cysteine instead, our sequence would just keep going as a read-through transversion mutation. Without a set baking time, our cake would end up overcooked and dry. Tens of thousands of mutations occur every day in every cell. Some are detrimental, but some are harmless. Without a proper recipe and directions and instructions, our baked goods would be inconsistent and at times inedible. Here are my citations. Thanks for watching.